Okay, I guess we'll start. We'll finish on what? On blood tissue. Then we jump quickly to DNA, to genetics, replication, transcription, those things. Then we finish with biotechnology. Hopefully by 9. Yeah, probably by 9. Then 9 to 10, Srina has said she will dedicate her time to tell us the answers to all those questions. Let me project my screen. So, hope you can see my screen. Divya, can you see my screen?
Yeah, I can see it.
How have you been?
I've been okay. How are you?
Blinking and breathing.
That's good.
Blood tissue. So according to the guidelines, the major thing we'll be focusing on is red blood cells. But as an overview, blood, when we think of blood, number one, we say blood is 75% or 60 to 75% water, right? Then and this water, we say water, or rather, we define plasma. No, the blood is made up of plasma and the cells, right? So we say plasma constitutes the 55%, which is, you can say it's the water component of blood. Then the 45% is the RBCs. Then the remaining, below, or rather, that 1%, usually the white blood cells and the platelets. So why is blood red? Because red blood cells are red. Right, that's a good explanation. Anyway, functions of blood, we'll just cover this in a gist. Number one, we know for transport, right? Transport oxygen from A to B, from your lungs to the tissues, and carbon dioxide from the tissues back to the lungs for excretion, hormones. In short, blood is where everything is transported. Don't need to mention much about that. Then regulation of number one, pH. We look at this once we are done with RBCs real quick. Regulation of pH. We said we mentioned about proteins yesterday, plasma proteins having an effect on the blood pH and such things. Regulating body temperature from high school. If you can remember, or even you personally, when it's hot outside, your hands are usually a bit more red. And when it's cold, a bit more white. Why? Because when it's red, blood nini. Because when it's hot, there is vasodilation of the vessel so that they can lose the heat. But when it's cold, there is vasoconstriction so that blood can retain its heat, right? Then regulation of fluid balance for protection, the white blood cells for clotting, right? We have all these functions of blood. Remember this diagram, plasma. If you, what is this called? Centrifugation. Yeah, this is how it would separate red blood cells because they are more dense. They'll be at the very bottom. Then at the very top, we'll have plasma. At the center is where you'll have your platelets and your WBCs. Then we say plasma is water is the major component of plasma. There's other proteins in it, and all these other components we've talked about. So let's dive to RBCs because they're the major focus of many of today. RBCs, red blood cells. When we think of red blood cells, we think of hemoglobin, right? It's the major constituent of RBCs, hemoglobin. What does hemoglobin do? Transport oxygen from lungs to tissues, and also transport carbon dioxide back to the lungs for to be excreted. One thing to note is that red blood cells do not have what? Red blood cells, number one, they do not have a nucleus, they do not have ribosomes, and they do not have mitochondria. So to take you back to day one of biochemistry, if they don't have a nucleus, that means it's not capable of replication, right? Because that's where the DNA is, right? If they don't have ribosomes, that means red blood cells are not capable of making their own proteins. Right? Then if they don't have mitochondria, that means they are not capable of producing energy through mitochondrial dependent mechanisms. Which are these mitochondrial dependent mechanisms? We mentioned on yesterday, number one, TCA, number two, beta oxidation, right? But the good news is that glycolysis takes place in the cytoplasm. Don't forget that because it's a commonly examined question. Glycolysis is cytoplasm, TCA cycle, the mitochondria. Yeah, then also they made in the bone marrow. We won't dwell so much here. All these modifications of no nucleus, no ribosomes, no mitochondria, you can, all these modifications is you can say to accommodate more oxygen so that it can transport it. Yeah. Then the other important point is that they generate ATP by anaerobic glycolysis. So remember, we said it only undergoes glycolysis. And what type of glycolysis? Anaerobic. RBCs, they are not able, as much as they transport oxygen, they are not able to utilize that oxygen they are transporting. Okay. Then regarding how RBCs are made, allow us to look at that when we are covering hematology and pathology. Even the synthesis of hemoglobin. Here, I'll just remind you there are, majorly two types of hemoglobin: the adult hemoglobin, the one adults have, and the fetal hemoglobin, the one the fetus has. So as adults, it's alpha and beta. 2 alpha chains, 2 beta chains. For kids, it's 2 alpha chains and 2 gamma chains. major difference between these two is that the embryonic, or rather the fetal hemoglobin has more affinity for oxygen than the adult hemoglobin. And it makes sense. Why? Because you want the mother to be able to transfer oxygen from her to the fetus. Yeah. Then there's also A2, that one is just in small amounts, 2 alpha chains and 2 delta. But remember, for the normal one, us adults, alpha and beta; for kids, alpha and gamma. So, so today we'll also cover, I don't know if it's here. Yeah. The why. What causes the fetal hemoglobin to have more affinity for oxygen than the mother's, or than the adult hemoglobin? And this is because of one thing called 2,3 biphosphoglycerate. Now, if I can take you back, it's in the next slide, that in glycolysis there was an intermediate such as 1,3-BPG in the steps of glycolysis. So this 2,3-BPG, as you can assume, it's formed from that. So 2,3-BPG, what does it do? 2,3-BPG, it makes, or rather, let's just say it decreases the affinity of hemoglobin to oxygen. Okay. So if I have a lot of 2,3-BPG, my hemoglobin won't bind as much oxygen. So simply put, you can say the fetal hemoglobin, we don't the adult hemoglobin has this there's the presence of this 2,3-BPG. Thus, it has a lower affinity for oxygen than the one for fetal hemoglobin. So take home message from this was just an overview. Take home message is blood. When you centrifuge, two components: RBCs down, plasma on the top. At the center, forming just a 1% white blood cells and platelets. RBCs major function, transport of oxygen. Something unique about RBCs is they do not have mitochondria, they don't have a nucleus, they do not have ribosomes, and the glycolysis is anaerobic. Okay. Then I wanted to cover more of RBCs, the metabolism. Is my screen the review of RBC structure? Is it there?
Yeah.
Oh, okay. So RBC is basic knowledge, by concave shape, right? Then we've read say no nucleus, no ribosomes, no mitochondria. So they obtain their ATP from glycolysis, right? And remember, anaerobic. Just keep remembering anaerobic. Then there's a unique thing about RBCs, and that is the capacity, or the unique ability to deform in order to fit in small capillaries. So imagine this RBC of yours. Yeah, this one here. It passes through arteries, so it can be like this, but eventually when it reaches your microcirculation, the vessels, they are not big enough to accommodate this size, so it has to, let's say, squeeze through, right? So that's the unique ability of RBC. It's able to squeeze through, or rather, it's the membrane. That membrane, it has sort of it can not deform, but more of adapt, right? And okay, we'll cover this more even we're looking at sickle cell disease and all that. Major function talked, won't go into it. It's transport of hemoglobin, transport of oxygen from A to B and carbon dioxide back. Synthesis, just remember it's from the bone marrow. The prerequisite, or not the prerequisite, the cell that comes before the RBC is the reticulocyte, right? Then now the reticulocyte has things like the mitochondria, things like the ribosome. Yeah, but for it to form that RBC, it loses that so that it can accommodate more oxygen or more what? Yeah, more oxygen essentially. Then a stimulus for RBC formation is erythropoietin. I hope we can recall that erythropoietin. And how would, now the question comes, what's the stimulus for erythropoietin? It's just a matter of oxygen. If oxygen in your body is low, erythropoietin is produced primarily from the kidney, but a bit from the liver, but majorly from the kidney. So when your oxygen is low in the blood and the kidney notices this and it's like, hey, maybe it's due to what? Low RBCs. So the kidney will produce more erythropoietin. That erythropoietin goes to the bone marrow and stimulates more production of RBCs. Then if your oxygen goes back to the normal or normal oxygen levels, they will reduce the production of erythropoietin from the kidney. So EPO, it's the major stimulus for RBC formation. The other stimulus is the thyroid hormone and testosterone. For the testosterone, just so something interesting is that before the discovery of erythropoietin, even ladies who had anemia and responsive to things like iron and vitamin B12, they'd be given testosterone injections because testosterone also stimulates RBC formation. And that is why you can also see guys have a higher hemoglobin level than ladies. So because testosterone stimulates production of more hemoglobin, of more RBCs. The necessary nutrients, we know them: iron, vitamin B12, folate. That's why types of anemia would be iron deficiency anemia, megaloblastic anemia due to vitamin B12 deficiency, and folate. Then Bagwat covered this yesterday, so I don't have to go through it in detail. RBCs, they have a lifespan of 120 days. 120 days is 4 months, right? And that's why they say you can't donate blood unless the last time you donated blood was more than 120 days. Or, okay, if you're a generous person and you want to donate blood every other time, give yourself 120 days, because that's the time it takes for, that's the lifespan of your red blood cells. So when red blood cells, they've reached that period, 120 days, they are broken down. Two things. Remember, red blood cells, when we mention red blood cells, always remember hemoglobin. It's the major constituent. So when it's broken down, we say we break it down to heme and globulin. Now, globulin, it's just a chain of amino acids. So that globulin, it just goes back to the amino acid pool of the body. But heme, we break down heme now to things like bilirubin. Yeah. So that is why when a lot of your RBCs are being broken down, bilirubin can accumulate, and you'll have things such as jaundice and all that. Again, anaerobic glycolysis, I won't repeat, no mitochondria. If I've mentioned something, I won't be covering it again. So metabolism of RBCs, how does it happen? We've said it undergoes anaerobic glycolysis. But how does it get the glucose? RBCs have GLUT1. Now, just to remind you, there are majorly 4 glucose transporters, GLUT1 to GLUT4. GLUT1 to GLUT3, they do not need insulin, right? The glucose will enter, they do not need insulin to open, but GLUT4, it needs insulin, right? So GLUT4 is majorly located in adipocytes and in muscles. Adipocytes and in muscles. Yeah, you'll notice that in the brain, we have GLUT1 and GLUT3 in the brain and in the neurons. So the brain doesn't need insulin to take glucose from the bloodstream, as again Bhagwat mentioned yesterday. So RBCs, GLUT1, they do not need insulin to take glucose from the circulation. So once they take that glucose, now, yeah, we go back to glycolysis again. Glucose to glucose 6-phosphate, all of that. Remember we said that one in glycolysis, in oxidation, let me say in aerobic glycolysis, we said the net yield of ATP was 2 ATPs, right? Because we are spending 2 ATPs and we are producing 4 ATPs, right? But you'll notice something. Okay, I'm not finding it. We'll see it in the comments. Like, I'm not seeing it here. Anyway, here it is, glycolysis. And here is the 1,3-BPG we've talked about, acted on by mutase to 2,3-BPG. Remember, we said 2,3-BPG, it decreases the affinity of hemoglobin to oxygen. So even Betty has ever asked the question in the exam that where do you expect 2,3-BPG to increase, the concentration of 2,3-BPG to increase? Is it when the red blood cells are near to the lungs or near to the periphery? And the answer is near to the periphery, because it's supposed to let go of oxygen there. When it's near to the lungs, you want it to have, you don't want it to have 2,3-BPG. You want it to take oxygen and to take it well, Now, you'll notice also here
Gaius,
that,
can you please repeat that?
That where do you expect the concentration of 2,3-BPG in RBCs to be at its highest? Is it at the lungs or at the periphery? Now, so the answer was at the periphery, because at the periphery is where you want the affinity of hemoglobin to oxygen to be at its lowest, so that it can let go of oxygen, so that it can give that oxygen to the tissues at the periphery. Then at the lungs, you want it to be as low as possible, so that it can have the highest affinity for oxygen, so that it can take up oxygen from the lungs.
Thank you.
Then again here, please just notice that glucose 6-phosphate here, we can see the pentose phosphate pathway, or rather glucose shunt. But the most important thing here, that take home message, is that we are seeing here this formation of what? NADPH. And as Seraphine yesterday said, NADPH, we need it to reduce this, in short, to reduce anything oxidative in our body, in terms of if it's this superoxides, because it works in this way. What reduces stuff, let me say, let's say, what reduces, for example, superoxides in our body is glutathione. But back to chemistry, if glutathione reduces something, it means glutathione gets oxidized, right? Now we need NADPH to convert the oxidized glutathione to reduced glutathione. So that means if we don't have NADPH, we won't be able to reduce this stuff. And Nani covered it yesterday quite well, so I won't dwell so much. So this is a statement we said in red blood cells, 2,3 binds to hemoglobin, decreasing its affinity for oxygen and helps its availability to tissues. Yeah. Oh, then is something here just unique. That in this step where we are generating NADH, it's used to convert with met, not here, what is it? Met hemoglobin to the normal hemoglobin, because met hemoglobin has no affinity for oxygen, and met hemoglobin is as a result of this iron 3 plus hemoglobin. In our hemoglobin, we want it to be iron 2 plus. Iron 2 plus is what is you can see it's the good hemoglobin. It's what we want. But the oxidized hemoglobin, that is iron 3 plus, not good. That hemoglobin does not like oxygen. So here NADH helps to make sure that we have our normal hemoglobin, not our met hemoglobin. Yeah. Then already mentioned this 2,3 BPG. So would go there again. Then pentose phosphate pathway. We've talked about the NADPH reducing the oxidized glutathione to reduce glutathione, which ensures that the superoxides are not hurting us. So just like yesterday, ribose phosphates is the end product. So glucose 6 phosphate deficiency, Nani covered it yesterday. That I don't know if there's a diagram. I won't dwell on this because Seraphine covered. No glucose 6 phosphate, that means NADPH, you don't have it in good amounts. That means you're not able to reduce these superoxides. That means what? That your red blood cells will lyse because of these oxidative elements. And before it lyses, it forms the Heinz bodies, because intracellular oxygens cannot be neutralized or cannot be reduced, and the globin denatures and precipitates as Heinz bodies. Yeah. So for glucose 6 phosphate dehydrogenase deficiency, it's what? It's it's something we should note with drugs such as antimalarials, because with antimalarials, let me just project that for antimalarials so that when we reach there, we'll have this summary, so even we won't have to. But with antimalarials, is there anybody in the audience that can remind us the mechanism of action of any, let's say, chloroquine? Chloroquine is one of the oldest antimalarial agents. What was chloroquine doing? Because once you understand what chloroquine was doing, you'll also understand why these increased why people in malaria areas have G6PD-H deficiency. In a gist, it doesn't have to be exactly at the mechanism as I want you to mention enzyme A, enzyme B. Anyway. Alright, let me just project that. Yeah. Here, let me so next week we'll be doing malaria. So this is this shout out to malaria. So when malaria infects your we know malaria targets RBCs. First goes to the liver, it's released and it hits our RBCs left, right, and center. So this malaria parasite, once it's inside your RBCs, it needs food, right? And what is food to it? It's your amino acids, right? Remember your amino acids, they are food. So the parasites will digest your hemoglobin so that it can obtain the amino acids. So that will mean just like what him we just like the way we break down hemoglobin, we make heme and globulin. Now this globulin, it has been digested by the parasite, right? So in this case, we just have heme. But heme, it has a high oxidative potential, right? Has a high oxidative potential. Meaning, if left unchecked, heme alone, heme by itself will do what? It can make the RBC lyse. Remember again, number one, no, nothing to reduce it. If NADPH is not sufficient, what will happen? The protein, the globins will be denatured, the Heinz bodies. But now in this case, we have heme. So what does the parasite do? Because heme will make the RBC lyse, and the RBC is the source of energy for this parasite. So the parasite is like, yo, my guy, what do we do? Let us make sure this heme is non-toxic to us. It converts that heme to hemozoid, right? To hemozoid. Once it converts it to hemozoid is not toxic. It's not toxic to RBCs. Hemozoid is just excreted. Yeah, it's put in a vacuole and excreted, right? So what did pharmacists come and do? They came in so that so the parasite, to protect itself, it converts heme to hemozoid. So if we make sure it doesn't convert heme to hemozoid, then it will attack us, right? So chloroquine comes here to prevent heme conversion to hemozoid, right? So if we've prevented heme to be converted to hemozoid, heme will build up. And remember, we said this heme part has a high oxidative potential. So that accumulation of heme will lead to what? The lysis of both the red blood cells and the parasite, right? To lead to the lysis of both the red blood cell and the parasite. Once that lysis occurs, the parasite has gotten hurt, but we've also lost our RBC. So heme, I want you to know that heme has a high oxidative potential, and that is why that is also the mechanism. It's the assumed mechanism of action of a drug such as primaquine. So with primaquine, with primaquine, it acts the same way. But for someone who doesn't have sufficient glucose 6-phosphate dehydrogenase. So they are not making enough NADPH to reduce it. That means that oxidative potential, it's not only dangerous to the parasite, it's also dangerous to the normal cell of this person. So that is why you'll see these antimalarials, not antimalarials in general, you know that term is wrong. Not antimalarials, but specific antimalarials being contraindicated for people with glucose 6-phosphate dehydrogenase deficiency. Then also with sulfur methoxazoles, the like of also with sulfonamides, the likes of sulfur methoxazole, sulfur containing not sulfur. These sulfonamides, they have also that oxidative potential. So even them, you have to ensure the patient does not have this deficiency of glucose 6-phosphate, because if they do, you risk doing more harm than good. Remember, something like cotrimoxazole can be given for a simple UTI. So yeah, it's something to watch out for. So okay. So looking at we already mentioned this methemoglobin reductase pathway. We said it's facilitated by this NADH. Why? Because I'm looking for the statement so that I can highlight it. Let's see. So here it is. Here it is. Methemoglobin, what does it do? It loves oxygen too much. So it doesn't let go of oxygen. So that is why we do not want methemoglobin, right? It's because it has a, oh yeah, I think I said a different thing, I'm sorry. It has a terribly higher affinity for oxygen, but this high affinity, it sounds good, but the issue is once it goes to the periphery, once it goes to the tissue, it doesn't let go of its oxygen. So yes, your RBCs have oxygen, but it's not letting them go. So what happens? No oxygen for your tissues, so you can die of hypoxia. Then briefly, we'll just look at the structure of the red blood cell membrane structure. It's made up of many different proteins. We can't look at all of them specifically, but just know it's made up of these proteins, and this protein is what gives red blood cells that unique ability to deform and penetrate microcirculation, penetrate those little blood vessels. Blood diseases involving the RBCs, anemia, we'll mention it more with pathology and even in clinical pharmacy. Then also this one, we'll mention them in the same place with pathology. So we won't cover that now. Sickle cell, the same. But now just for sickle cell, remember, before sickle cell, we've seen 3 types of hemoglobin, right? We've seen hemoglobin A, the one normal in adults, right? We've seen hemoglobin F, the fetal hemoglobin. Then we've seen hemoglobin A2. It's also in us, in adults, but in low concentration, just a little, like 2.5 to 5%. Now, hemoglobin S, that is now sickle cell disease. Now, that is another type of hemoglobin. If you have this type of hemoglobin, you have the sickle cell disease. And it's due to when you replace glutamic acid with valine. That replacement, that amino acid, is what hurts you. And essentially, Okay, what happens with sickle cell disease is the issue is at the cell membrane, right? We haven't dug deep into the cell membrane and its constituents, but just know that there's a diagram I'm looking for that will just summarize that. The issue is at the cell membrane. Uh, uh, it is, let me project that. So, so because even what we'll cover is we'll cover it further in clinical. Take-home message of RBCs is number one, it doesn't have ribosomes, it doesn't have mitochondria, it doesn't have a nucleus. Okay. Number two, it undergoes anaerobic glycolysis, and because it undergoes anaerobic, not because it undergoes anaerobic glycolysis, how does it get its glucose? It gets it from the blood, but it has GLUT1 transporter. In short, it doesn't need insulin to take glucose from the blood. Okay. Then we've also mentioned there is the Rappaport shunt pathway, whereby the RBC generates what? 2,3-BPG. Why is 2,3-BPG important? Because it makes sure that it reduces the affinity of hemoglobin to oxygen. It sounds like a bad thing, but it's actually a good thing when that red blood cell reaches the periphery, or when it reaches the tissues where it needs to let go of the oxygen. And we've seen that in the adult hemoglobin, we have more 2,3-BPG than in a fetal hemoglobin. That is why the mother is able to that transfer of oxygen is able to happen. Then we've gone ahead and we've seen also methemoglobin. And methemoglobin is just, instead of normal hemoglobin has iron 2, but methemoglobin is iron 3. Iron 3, it's so iron 3, it's methemoglobin. It has way too much affinity for oxygen. That means even when it reaches the tissue, it's not able to deliver the oxygen to the tissues. It holds on to that oxygen, and thus tissue hypoxia happens, and you can imagine what happens next. Then how is this how is glycolysis helping with this? Glycolysis generates the NADH, which essentially it converts iron 3 plus to iron 2 plus, making sure that you do not have methemoglobin. Then we've gone ahead and seen also that the red blood cells also do take, there's also the hexose shunt pathway, or the pentose phosphate pathway. And we know there's one major thing we get from the pentose phosphate pathway, that is NADPH. Why is NADPH important? It reduces oxidized glutathione to reduce glutathione, which reduces any oxidative species that may be in our body. So if you have G6PD-H deficiency, you are at risk of this oxidative species hurting you, causing hemolysis of your red blood cells. And that is why we've seen the Heinz bodies, right? When your globulin part of hemoglobin is denatured and it forms those precipitates. And then I think that is majorly it. That is what we've seen. And with sickle cell, I'm not seeing what I wanted project. It was a really good photo, but I'm not seeing it, but we'll have it next week. The other week when we are doing hematology, we'll cover sickle cell disease then. So if there are any questions, please ask. We'll take a 7-minute break at 8:20 to probably just 8:40. We go through genetics. We'll focus on transcription, replication, and translation. Then we go directly to biotechnology, where we'll just highlight the key things. Then Shrina will project the question papers. Divya, are we good?
Yeah, we're okay.
Yeah, okay. So we can start DNA. We can start genetics.
Yep.
Happy to hear it. Happy to hear it. Let me ask Tevin, are we good?
Yeah,
Nice, nice. Gender equality, gender equality. So DNA. I for this, I couldn't find some good notes, so if someone has the good notes or something. But okay, this book is summarized. It's mostly pictures, so yeah, hoping it will help us out. So genetics, yeah, study of inheritance patterns and such things. We know what genetics are, right? Genetics has to do with genes, you know, someone has genes for this, genes for that, whatever. But at the very center of genetics, or at the very center of genes, at the very center of inheritance, what makes you have, what makes you your sister, your brother's brother, whatever. I don't know. I'm not a pastor. But what you share with your family is some common genetic, let's say some common genome, or rather you share DNA. That's what makes you biologically a relative, right? People share DNA. That's what we say: biological father, biological mother, brother, and all that, right? So DNA is at the center of everything. And we can say DNA. So because, for example, when people are tall in a family, we see that even generations ahead, they go ahead being tall. That means something in their DNA is making them tall. Same with a disease, right? We say something like cancer, we always have to look at genetics. Something like even the likes of CKD, hypertension, we say there's always a chance that there's a genetic component because you may inherit this, even sickle cell. Yeah, there's a chance of inheritance. That's in short. And at the root, it's DNA, nucleic acid, deoxyribonucleic acid. So deoxyribo, that's just deoxyribose. That's a sugar, right? A 5 carbon sugar. Deoxyribose, and then nucleic acid, that tells us there's an nucleic acid attached to it. And then silently, there's always a phosphate. Wherever there's a sugar and there's that nucleic acid, always expect a phosphate. Then there's the question that is usually asked: what is the central dogma of biology or of molecular biology? Simply, information flows from DNA to RNA to proteins, right? That means, for example, for people yeah, let's say the sickle cell disease. Let's work with that, because the good thing is that we will see sickle cell when we are talking about biotechnology. So let's give that example. Sickle cell disease, right? The issue is at the DNA level, right? That there's something there's a membrane protein that they are not forming well, right? So it comes from the DNA, right? That instruction comes from the DNA and it's transcribed to RNA. RNA goes to the ribosome and it's translated to make a protein. And now the protein is the one that is defective, right? Because the protein is defective, now this person will have sickle cell disease. Same with let's say now in the case of this. Okay, it's going away. No, yeah, in the case of the COVID vaccine, right? The COVID vaccine were RNA vaccines. So RNA vaccines, so that rumors, those rumors that this Nini changes your DNA, the COVID vaccine changes your DNA. It's okay, according to what we know as of today or currently, according to what we know from molecular biology, is that RNA is translated to form a protein. That protein is now the what? The antigen, right? The antigen of COVID. And that antigen is what will teach your immune system to fight COVID-19. And there are a lot of RNA vaccines. We'll see them again when we are covering, I think, immunology. We'll look at how vaccines are made and all that. But just know that the central dogma of molecular biology is that information flows from the DNA to the RNA to the protein. So the DNA contains the encoded information that this person will be told. Maybe a lot of growth hormone, a lot of long bones, things such as those. That information is transcribed to RNA. RNA, we form proteins that will enable this. Yeah. So looking at the structure of DNA, here it is. We've said DNA is deoxyribonucleic acid. Now, nucleic acids, we have, I don't know if it was covered. We have the pyrimidines and the purines. Yeah, purines and pyrimidines. I don't know if it's here. If it's not here, okay, we went to check. Purines and pyrimidines. Now, for purines, we have adenine and guanine. Then all the others are the pyrimidines. Just remember that. Purines is just adenine and guanine. All the others are pyrimidines. Now, what the structure of DNA basically is what you need to recall is that you have, here is your purine or your pyrimidine. Now this is a pyrimidine. It's linked to the sugar. Here is your deoxyribose. And then this is linked to this phosphate group. Now that phosphate group links it to another sugar, right? So in short, know that the sugar is linked, the nucleic acid, whatever it is, adenine, thymine, guanine, whatever it is, the nucleic acid is linked to the sugar. Then the sugar is linked to the phosphate, and the phosphate links it to another sugar, right? So what holds DNA together? It's this phosphodiester bond. Phosphor, why? Because of this phosphorus. Diester, why? We are seeing O diester bond. You don't just know phosphodiester bonds. It's what holds DNA together. It's what links the your different DNA, your different nucleic whatever. It's what links your DNA together, basically. And then on that note, adenine links with guanine. Here, where it is it? You remember, I think we remember that rule. Adenine goes with guanine. No, it's not guanine. Adenine goes with thymine. Cytosine goes with guanine. That's why, just remember that. Adenine type ATGC. You can just ATGC. That's how they are interlinked here. Now that diagram is not right here, because we know DNA is double-stranded, right? So in this strand is A, it's linked to the other strand by T, ATCG. And I want you to note something here, it's because it's not a mistake. A and T, we are seeing two lines, two bonds. C and G, 3 lines, 3 bonds, right? So essentially, CG, that CG pairing is stronger than AT. Again, that's a common question. Between C and G or A and T, which one is stronger? It's C and G, because there we are seeing it's linked by those 3 hydrogen bonds, while AT is just 2 hydrogen bonds. So yeah, so that's DNA for you. DNA is double-stranded. It's made of a sugar backbone, right? Here's the backbone. It's made of a sugar backbone. The molecules are, the nucleic acids are interlinked with the other strand in this manner: A to T, G to C. Then we always say that, or rather, we look at that when we're going through replication. We've already reached. We've talked about base pairing AT, CG. Yeah. Now let's look at how is DNA replicated, right? How is DNA replicated? And let me just project this one. I find this picture happening. Stay with. So DNA as a, or rather as a summary, number one, it's we know DNA number one is double-stranded, right? DNA number one, it's double-stranded. So the first thing we want to do is break that double strand so that we can have it as single strand, because it's hard to replicate something that it's wound up on itself, you know. So the first thing we have is this enzyme called, and this is what will summarize replication. This is just what we are covering here. First thing is we are having DNA helicase, which will go breaking the hydrogen bond. So in short, it splits the 2 strands simply. DNA, and remember this, DNA helicase, it's the first enzyme for replication. It's the first one. It breaks it and splits. It splits the 2 strands into single strands. And now in that case, there is now a strand that this one, it's in a 5' to 3' direction, and this one is in a 3' to 5' direction. But we will cover that when looking a bit. Okay, we'll see. Well, okay, so DNA helicase, what has it done? It has gone and it has splitted the 2 strands. But what do you expect? Just picture it in your mind that here, going down here, if we just split one area, then down there it will coil more. I think we've all encountered this with earphones, right? When you try to separate earphones, at the very end, that's where it's more, it holds together more, right? And now in this case, that is what we call supercoiling, right? So, and this supercoiling, it can lead to breakage of DNA. That's supercoiling, because when you supercoil too much, then it will snap, right? And this is where topoisomerase 2 comes in. Topoisomerase 2, as you can see, here is your DNA helicase. Then topoisomerase 2, it will go ahead of the DNA helicase to relax that supercoiling. It makes sure that supercoiling does not lead to breakage. Okay, it goes ahead of it. Then now we have the single strand, right? A single strand. Then there are these proteins that will be on the single strand, the specific single strands, to keep them apart, so that now replication can take place. And what replicates the DNA? It's the DNA polymerase 3, or just DNA polymerase. It will make the new DNA. And know that DNA is made in a 5' to 3' direction. Just memorize that. Everyone, just DNA is made in a 5' to 3' direction. Always. Even when we are looking at this other strand, we will see DNA is always made in a 5' to 3' direction. Why? I don't know. So memorize that. I'll repeat it one last time. DNA is made in a 5' to 3' direction. So here's your DNA polymerase, right? Or is it here? It has moved from here where this is why we call it the origin of replication. This is where replication is start. It's making DNA because the parent strand, we call this strand the parent strand. And now the new strand is the daughter strand, right? So this daughter strand here, because the parent is in a 3' to 5' direction, then the daughter will be in 5 to 3. So this leading strand is what it will be synthesized in continuous manner. I want you to note that this one, from where the RNA primer binded, we can see here it's gone on very well, very smooth, right? Now and that is what we call the leading strand. Now let's look at the lagging strand. Now in this lagging strand, you can see the parent strand of the lagging strand here is in a 5' to 3' direction. And that means we can't make our DNA like where is it? Let me annotate. That means we can't make our DNA going how? Going this way. Going this. It's confusing, but okay. We can't make our DNA going in this manner, because in this manner it will be okay. DNA again, I'll just repeat because my laptop is acting up. It's made in a 5' to 3' direction. So here, how we will make it is that we will be going down like this. We make a small strand, and then we come here, make a small strand, and then we join them together. In short, we make them in form of fragments. Then this DNA ligase is what will join the fragments together. But so know this. DNA helicase is the first enzyme. It goes and splits. It splits the 2 strands. Now, and then topoisomerase 2, because once it splits the 2 strands, there'll be supercoiling. So topoisomerase 2 relaxes that supercoiling. Then DNA polymerase begins its work synthesizing DNA in a 5' to 3' direction. So for the parent strand that it's easy, it will make it continuously. But for the parent strand that is in the opposite way, it will have to make it in fragments. And then how will we join these fragments at the very end? We will use DNA ligase. Simple as that. So that's replication. Then so take-home message of replication is those enzymes. First enzyme, DNA helicase, breaks the 2 strands. Topoisomerase 2 goes ahead and relaxes the supercoiling. the DNA polymerase replicates the DNA in a 5' to 3' direction. And then the DNA ligates join the fragments. We're a bit behind schedule, so I'll just go directly to RNA. How do we make RNA? Because you said information flows from DNA to RNA. So first, the major difference between DNA and RNA is that RNA does not have thymine. Right? It doesn't have thymine. Instead of thymine, RNA will have uracil. So wherever there is, if you are told, here it is, if you are told to make substitute the sequence accordingly, wherever there is, for example, here, the DNA, yeah, if you've been given that RNA sequence, know that RNA does not have T. So A will be with U, but G will remain with C. Then there are 3 types of RNA. There's the transfer RNA. I think we know about the messenger RNA and the ribosomal RNA. So messenger RNA is what we will make from DNA. Transfer RNA is what will bring the amino acid that we will use to build the protein. Ribosomal RNA, as it in RNA, is just a complex in the ribosome that is involved in protein synthesis. Yeah, there's somewhere down here. So okay, while I look that picture, when it comes to RNA, we will have first of all, we will define one thing. We know DNA is long, right? DNA is very long, but there's 2 parts to DNA. That is or rather to a gene that will help us just learn RNA in an instant. Yeah, here it is. So a gene in your DNA, a gene is just something, or rather the sequence in your DNA that is used to make a product. Like in short, something that will be used to make a product. That's what we call a gene. Sometimes we'll say something that will make a protein. Now from the genes, we transcribe the genes to form the RNA, right? The messenger RNA here, transcription of this gene produces an RNA. This, we call it the messenger RNA. And then the messenger RNA is what will go to the ribosome and will be translated to a protein. The protein is what will conduct the function. Now for transcription is where we have a few terms that now we need to memorize. So number 1, we have, let me see, let's cover it as this. That there are 3 stages of transcribing a gene. The first stage will be initiation, whereby we look at the promoter region. The second is elongation, whereby you're just adding the nucleic acids to the RNA. And the last thing is termination. Termination is unique because we'll see there's a termination sequence, because how we'll transcribe transcription know when to stop. And so here we'll cover in simple terms. I hope you covered this when you are reading before, because we won't go that much. So, initiation. With the 3 steps: initiation, elongation, termination. The initiation step. What happens in the initiation step? Number 1, we said we have a promoter region. Now, in the promoter region, we'll have transcription factors, which will, like, these transcription factors is what will enable that gene to be transcribed to messenger RNA and finally to a protein. Remember, example of not even an example of a transcription factor. Let's look basically at something like estrogen. Estrogen enters the body, enters the cell, binds to its receptors, and that complex moves to the DNA, to the nucleus, whereby it acts as a transcription factor. So what would you expect estrogen to work to do when acting as a transcription factor? Now you're expecting that, for example, there'll be more what will be maintained, the functions of estrogen. That the vaginal environment will be well maintained, well lubricated. That's why in menopause, these things, there'll be this dryness of the vagina. So essentially, how estrogen works is that it binds to its receptor, then acts at the DNA level, enabling synthesis of things, the results. Okay, it binds at the DNA level and enable synthesis of proteins that will serve its function. So, and that is what we need in the initiation step is this transcription factor, which will now enable RNA polymerase to bind to the promoter. Remember, when we were looking at replication, we saw DNA polymerase. That is what makes DNA. Now here we are looking at making RNA, and here we are seeing RNA polymerase. So once RNA polymerase binds to the promoter region, it will just synthesize DNA, synthesize the messenger RNA. Then finally, that at the end of that gene, it will stop. It will reach somewhere and it will stop, and the RNA will be complete. The RNA will now move to the cytoplasm, where it will meet the ribosome and be translated into a protein. Now, for all these parts, because of time, just go through it for yourself. But know that those 3 steps. Number 1, initiation. Number 2, elongation. Number 3, termination. Then there's also, once that RNA is made, there's also some modifications that is made to the RNA molecule so that it can reach the, here it is, this table. This table should be enough, so that it can be functional. So number 1 is, if it's a very large RNA that has been made, it can be split into smaller pieces. Then number 2, there's these 2 things. Even Jonathan said they like asking in board exam. This is just what I wanted us to look at. Number 1, there is this 5 capping and poly A tail. So the 5 prime capping in A, what the role it plays is just in facilitating the splicing of introns, the exit of mRNA from the nucleus and the binding to the ribosome. Then the poly-A tail, it is important for the stability of the RNA and translation in eukaryocytes. The stability of the RNA and translation in eukaryocytes. So the 5' capping, know that exit of the RNA from the nucleus, splicing of introns. What are introns? Introns are the parts of the RNA that will not lead to synthesis of anything. There is introns and there is exons. Exons are what we'll actually use to make the protein. Introns will not make anything from it. So remember this 5' capping and poly-A tail. Poly-A tail, stability of the RNA and enabling translation. 5 capping, exit of the RNA from the nucleus, and binding of the RNA to the ribosome. Okay, that will be it for this. For replication genetics. But take home message from, again, just to repeat, from DNA, remember the enzyme. DNA helicase splits, topoisomerase ensures or rather relaxes the supercoiling. DNA polymerase synthesizes the daughter strands, and this scrub, the replication happens in a 5' to 3' direction. So for the strand that it won't happen continuously, we go ahead making fragments that we will join together using DNA ligase. Yeah. For the translation, we'll see translation even when we are covering things like mechanism of action of the likes of tetracycline. So yeah, unless there's any questions, additions, comments, I don't know. Seraphine, is she back? Or Seraphine is not back. Sanaipe, questions, additions. Or comfort, comfort. It's been long. Let us hear your voice at least.
Yes, hiyo
Anyway, yeah.
nini
As you
kuna
can see, even as summarize here, when it comes to
okay, poly-A tail.
sijui kama it's my net, sija kusikia Jambi.
It's your net but nimeona ni poly-A tail na hiyo 5
Mungu.
cap.
Yeah. Those are the major ones. Yeah. Yeah, so here it is again, addition of that 3' poly-A tail and the 5 capping. And also now for that removing of the introns and joining of the exon. This is example of those post-translational modification that happens to the RNA. So when you have time, just go through. I think Seraphim shared the book, so you can just go through it. And finally, in 10 minutes, we'll cover pharmaceutical biotechnology. Not like we have a 10-minute break. Like we'll cover it in 10 minutes. That was. So biotechnology, who remembers biotechnology? Anything from biotechnology? Jambi, now that umeongea.
Yeah. Sijui, labda nikumbushe.
What's the most common drug that we use?
The most common drug we use.
Yeah, that is in use. Let me say that is in use. That is made. That is as a result of biotechnology.
Ni mingi. Like zinaitwaje? Hizi okay, sijui the most common, lakini tuseme hizi kina nini zinaitwaje? Okay.
Monoclonal.
Oh, antibodies.
Antibodies. Ni hiyo ulikuwa unatafuta.
Nilikuwa nataka kusema hizi hormones kina nini? Hormones hizi kina levonorgestrone, but I don't know.
Okay, kuna hormone, one hormone that is, I think it's the most common.
Insulin.
Insulin, insulin. Yeah. And now, because of that, we'll just be covering major. Or rather, we'll summarize biotechnology using insulin because it's the most common, as Jambi has said. This diagram, yeah, this diagram. Right, so here, just as a background that you yeah, as a background, yeah. Biotechnology essentially, or rather pharmaceutical biotechnology. When pharmaceutical, we mean, here we know that means drug. Bio means life. Technology means, I don't know, tech. So it means using technology on living organisms to make drugs, right? Using technology on living organisms to make drugs. And we said for this one, we'll majorly just be using insulin. But now the basics before we go to that is there are just a few definitions, such as what is a plasmid, because we'll be seeing a lot of plasmid. What is a plasmid? What is a vector? Then once we've understood that, we summarize. Not even once we've understood. Once we've stated that, we can summarize the whole. So plasmid. Let's just look at plasmids first. Plasmid, from this diagram here, you can see a plasmid is essentially an extra chromosomal DNA molecule, because it's here. Here is the chromosome, but here is a plasmid. It's found here in the cytoplasm. So it's just a DNA molecule, but it's not bound here in the chromosome. It stands on its own, right? And through plasmids is how resistance is usually spread, because you can see, for example, let us say this is E. coli that is resistant to nitrofurantoin, right? And that we know that resistance will be encoded somewhere in its DNA. Just like the mechanisms of resistance, we remember something like making an enzyme to degrade what? To degrade the drug. So even, let's use a better example. Let's use Staph aureus, because it's a common one. Staph aureus, at one point in time, it used to be killed by penicillin. But then what did Staph aureus do? It went and made beta-lactamase. Beta-lactamase essentially breaks down that beta-lactam structure and making penicillin not touch Staph aureus. So I want you to imagine this is Staph aureus. And for us to make an enzyme, as we've done from biotech, an enzyme, we said enzymes are proteins, right? And central dogma of biology, information will flow from DNA to RNA to proteins. That will mean that information to make beta-lactamase is found somewhere in the DNA that will be transferred to the RNA that will later be transferred to form the actual enzyme, beta-lactamase. So I want you to imagine that information is also in this plasmid. So this Staph aureus here is resistant. This one is not resistant. You see? This one, let's say, does not have that plasmid. But then it finds its good friend. This conjugation happens. They link through a pilus, and this cell is able to transfer a plasmid to this other cell. And now this cell is also now resistant to that drug. That's why we say antibiotic resistance can be spread, because bacteria, they can share that information. Okay, it's impossible to look at biotech even in a few seconds, but I will summarize it all in insulin, as we say. There are other ones, but will be guided by the questions that I brought. So, how do we make insulin? How do we make insulin from bacteria? First of all, we make it as humans. We make insulin in our pancreas, right? That will mean that information for making insulin, that gene, that sequence is found somewhere in our pancreatic cells, specifically in the beta cells. So we go to our beta cells and we isolate that DNA sequence or that gene for making insulin. And once we isolate it, we bring that gene and we put it here. We insert it here, as you've seen. We insert it, and then here it is back in the bacteria. We put the bacteria in a media which has the essential growth components, and we know because it's in the right environment, it can grow, right? Or it can grow and it will express its proteins. And which are these proteins? It's that insulin we've embedded inside of it. So that bacteria will be able to give us insulin. But remember, this insulin, we need it to be pure. So we will also purify that insulin before packing it and now giving it to the client. Just a few things to mention there will be, number one, there are two types of enzymes, two classes. Let me not even say classes. Let me use the word, but yeah, I think types was the right term. Yeah, two types of enzymes that we use in biotechnology, because you'll ask the question, why, how do we cut DNA and all that, and it will be a tough question to answer. But there is one thing called restriction enzymes, and the other one is called the, not ligase. Is it ligase?
Yeah,
Or
it's
is it
like
here?
this.
I think it's ligase. Oh, it's ligase. Okay. Yeah. Yeah. Oh, yeah. It is the restriction enzyme and the ligase mix the join by base pairing. Oh, it's not written here. Okay. Yeah. Remember, we've seen DNA ligase before when we're looking at replication, joining of the fragment. Now, so the restriction enzyme is what we use to cut. It's what we use to cut. Now, imagine we have your DNA. The restriction enzyme is what we will use to cut the part we need. So that tells us the restriction enzyme has to be specific. You know, it's not one restriction enzyme fits all. The restriction enzyme is specific, so we use it to cut my DNA, the beta cells, the part that synthesizes insulin, we use it and we cut. And then we go to the bacterial plasmid and we cut the part where we want to insert that insulin synthesizing gene. Once we've done that, now we want my gene and the gene of bacteria to mix. Now we will use ligase. Ligase will join the two like this. It will join the two. Now they are one. Once it's joined together, now the bacteria, we culture it or fermentation, and now the bacteria will give us all the insulin we want. So in this case, then the other part that I'll just briefly look at is antibodies. Just how are the antibodies produced? Then Shrina can project. Because there are majorly two types of drugs that are produced. The the proteins, and now the monoclonal antibodies. If anything, the monoclonal antibodies are the major ones that are produced these days. Just look at that. Yeah. So let's use this diagram to summarize. So how do we produce the antibodies? Okay, this is polyclonal. That will just, okay. There's one for monoclonal. Don't want to be misleading. Yeah, it is. So to produce the monoclonal antibody, first of all, we inject, let's take the example of we are producing a vaccine for COVID. Is just a basic example like that. So number one, we'll isolate the antigenic portion of COVID-19, inject it in this mouse. Then the mouse, naturally, it will produce antibodies against that antigen, right? And okay, this is a bit of immunology. How do we produce antibodies? It's once the antigen enters your body, it is identified by the, or rather it binds to that B cell, and the B cell separates, or rather let me say processes it. For example, we've injected the mouse with COVID-19. Let's just call it COVID-19, but in our mind we know it's an antigen. We've injected our mice with COVID-19. That COVID-19, the B cells will see it, engulf it, and analyze it. Look at, once it analyzes it, it will produce, or rather, it will produce two types of cells from it. One we call a plasma cell, and the other one we call a memory B cell. The plasma cell is what will produce antibodies specific to the COVID-19. The plasma cell is what will produce the antibodies that are specific, that will now bind to COVID-19 and kill it, or rather will opsonize it, right? So keep that in mind. So we've injected this mouse with COVID as shown. Here are the plasma cells. These plasma cells, we say they are the ones producing antibodies specific against this COVID-19. So we merge these plasma cells with tumor cells. Specifically, we call it myeloma cells. Why myeloma cells, or why tumorous cells? Because they have an unlimited capacity of division. So we merge them together to form this hybrid cell. We call it a hybridoma cell. Now, these plasma cells, because they are producing antibodies and the unlimited capacity of this one to replicate, they will just be producing for us these monoclonal antibodies. These monoclonal antibodies, now, for example, something like rituximab, or the one for breast cancer, trastuzumab, right? Now that is what we will go and use in the clinical setting, that monoclonal antibody. But remember again, it's not as direct as it seems, because it has to undergo steps such as purification, isolation, because it's not as if this hybridoma cell will be producing specifically monoclonal antibody. There'll be contaminants, because remember, it also has products of its own metabolism and such things. So yeah, about monoclonal antibodies, that is it. Just memorize this diagram. It will really come in handy. That's it, unless there are any questions. The other part, I encourage you to go through it, especially the DNA replication and transcription. Just repeat it. Let me stop sharing. Shrina, the floor is yours. It's 9:13. Or anyone who will project the screen for the questions. And now we can discuss. Now, Sanaipei, Faith, Comfort, Janet, Kipunja, Divya. Divya, even you can read the questions for us.
Is it starting from 59 or? Okay. All of the following are examples of proteins with the principal function of, what's that? Yo, what's that? Principal function of dash in the body except. Now, what's the dash?
Transport.
Oh, transport in the body except fibrin, as we said yesterday. And then a polar amino acid from the given list is glutamic acid. I think we did this yesterday. Mhm. I've seen these for if you guys want questions starting from what we've done today, there's 78 of one of those two papers. One of those papers there. I don't know if you want me to project. Mhm. So, so most naturally occurring sugars belong to which configuration? I think his answer is right. Or guys, what do you think?
I think it's L. I read in a textbook that most of them are in L form. In D form, they're very rare. I may be wrong.
From Betty's notes, it says D is the most common.
Yeah. Because even glucose is the
D form, yeah.
yeah.
Or it was amino acids. Just a minute, let me check.
Mm-hmm.
Okay, it's D amino. In amino acids, most of them are in L form. D amino acids are often found in polypeptide antibiotics. Sorry.
Thank you, Shireen. So in amino acids, it's L.
Yeah, you guys can see my screen.
Yeah, we can see.
The one I'm projecting for the notes.
Yeah.
Yeah. Hey, you've highlighted the whole book. Olivia, I hope you're seeing what other people are doing.
Yes, I can see that.
Take it easy on her.
It's okay, I'm used to him. Okay, so for a gene to be expressed, it must be transcribed into mRNA, right?
Yeah.
And then in the
You're right.
DNA, double helix guanosine is usually paired with cytosine. Okay, guys, I feel like I'm talking to myself.
That's the answer.
Yeah.
Okay, so in carbohydrate metabolism, the lack of dash results in diabetes mellitus. I feel like this guy knows all his answers,
No.
right? Okay,
Yeah, this guy knew everything, but it's even stressful.
that'll be you guys. Don't worry. Eh?
Hey,
Okay.
I don't know anything, but I ask Patrick.
Which of the
Can you
following
shut up?
compounds?
Can you shut up? You know a lot.
Okay, Patrick, we'll get there. Okay, so which of the following compounds is not a ketone? Patrick, tell me the answer.
You're glyceraldehydes.
Mhm.
Glycerin.
Yep. Okay.
Yes, in
Nini?
that's an aldehyde, yeah.
Mm.
Yeah.
The the principal function of essential fatty acids in humans to serve as precursors for.
Yes, it's hiyo ni normal.
Mm.
Fatty acids in human. Proteins ni amino acids. Enzymes zinaformiwa na nini?
Enzymes are also proteins.
Ah, okay. Then vitamin
The different enzymes, I guess.
Not okay, vitamins.
Essential fatty acids. What's an example of essential fatty acid? Guy, I feel like I'm forgetting.
Is acetyl-CoA no? Mm. All those things that have, I don't know,
Omega-3 and omega-6 from according to Google.
oh,
Yeah. There is linoleic acid, the alpha linoleic, omega-6 and 3. So
okay.
they regulate. Okay, so these fatty acids make hormones. So I'm saying prostaglandins is hormone. Is a hormone. Okay, I don't know. Guys,
I'm seeing somewhere that it's saying they are essential for making vitamins, cofactors, and yeah, so also prostaglandins, thromboxanes, lipoxins are in the list. So I don't know if it's vitamins or prostaglandins.
so mendana prostaglandins.
I guess. Or vitamins. Prostaglandins or vitamins, according to what Jambi has said.
Mm.
It has to be prostaglandins because prostaglandins are made from arachidonic acid, which is a fatty acid.
Okay.
So, yeah, I think that's the answer. I'm not sure.
But are vitamins do we manufacture?
Patrick, what?
I'm asking, do we manufacture vitamins? See, they come already made.
I think
You
like the liver does something. Does it produce some vitamins? I'm not sure.
I think it does because the supplements are used when you have deficiency. Ama?
Yeah, that makes sense.
Yeah, that's what I'm saying. When you kula whatever you are kulaying,
Yeah.
sinakwanga already hizo vitamins zisha nini. Sorry, the vitamins are already like we consume vitamins. I don't think we manufacture them in the body. I mean, we can convert them to their active form, but I don't know if we manufacture.
Okay, I guess we'll come back to 82. Or when you're reading for yourselves. Let me go to number 84. Triglycerides are solid at room temperature and are usually referred to as fats.
Right.
yeah.
Okay, just give me a minute, kindly. The major lipoprotein source of cholesterol used in cells is LDL, isn't it?
Yeah, because
In the
okay.
reaction, ATP plus glucose will give you ADP plus X. What is represented by X? What we just did in glycolysis, or
Because 6-phosphate.
and
Answer is
then
okay.
yeah, which of the following compounds is regenerated at the end of TCA cycle? That's oxaloacetate. And then which of the following is not no, not that one.
Yeah, we need to know even the sampling method. Wow.
Lazima. Haya.
Kimeumana, kimebite each other.
I guess we can also
But
do
you
the
know,
questions that were in chem. Sorry. Hmm?
These ones are coding organic chem.
Then I think biochem is over from this paper. Mama?
It's flashing now.
Okay, I've seen a question here. Number 66. I don't know if it's the same paper as Shrina's. Shrina, do you mind if I project my screen? All right, thank you. Okay. I hope you guys can see my screen. All right, so all of the following are sources of nucleic acids required in recombinant DNA technology, except
Which number are you reading?
number 66. Can you guys see my screen?
Yeah.
Yeah, I'm reading number 66. So all the following are sources of nucleic acids. What I don't get is why is there bacteria and bacterial plasmids? Can't it be, I don't know.
Can't it be?
All the following are sources of nucleic acids required in recombinant DNA technology, except. So there's human blood, there's viruses, there's bacteria and bacterial plasmids. From what we've just done, bacterial plasmids is there. Bacteria is used for you to put in your medium, and then it grows, it cultures. So I don't know about these two, human blood and viruses. Of course you use, I don't know. What do you guys think?
But I think like maybe the same way they were taking a sample from a rabbit, maybe you can replace it with a human because you'll need it to make the antibodies.
Mm-hmm.
So I think human blood is there. I don't know. I'm not sure.
But I feel like
But
even
viruses and bacteria can like
yeah. But it says sources of nucleic acids. It says sources, so I don't know.
But the blood has nucleic acid.
Mm.
Okay.
I feel like I'd go with bacteria, but also viruses could be there, but I'm not too sure. Anyone?
Anyway, let me try and Google the question. Okay, so the core. So I think nucleic acid enzymes like. So I think the answer should be human blood because viruses. Anyway, let me just read. Nucleic acid enzymes such as DNA polymerases, RNA polymerases, reverse transcriptases, DNA ligases, etc. Plasmids and bacteriophages are the core elements for recombinant DNA. So we can use enzymes isolated from DNA. We'll just come back to it, Basi. But me, I'd have gone with
Human blood.
with 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 viruses.
With viruses, okay.
Okay, so I think human blood, because it's not a source of nucleic acid that is required. Even the textbooks mostly talk about viruses, bacteria, and plasmids.
Okay, so I guess we can just put like a star here. Right. So the following are gene cloning vectors except there's plasmid, bacteriophage lambda, bacteria, artificial chromosome, and cosmid.
Choice C.
Why?
Elimination.
The flying, huh?
Yeah.
But what does this do? Artificial chromosome. Bacterial artificial chromosome. Yeah. What's a cosmid? Wait, let's see. I want to search.
Cosmid iko. Cosmid.
So what is cosmid?
Let's see.
Yeah,
Let me just
yeah,
search.
cosmid and plasmid are there. Then
Cosmids are plasmids. Eh?
Hybrid plasmid that contains a lambda phage cos sequence. Okay.
That's crazy.
So streptomyces bacteria have what the hell? Have been used in technological production to make one of the following drugs. So there's gentamicin, paracetamol, streptomycin, and morphine.
Streptomycin.
Are we just going with the answers that are there? Anyway,
Well, the name
That's
is
that's
streptomycin.
Saccharomyces, or what's that?
what?
What?
The 68, is it Saccharomyces? Or what is that?
Kuna
Like it,
gentamicin,
no,
paracetamol.
like what is
Paracetamol.
the question? Yeah, there. What is that?
Streptomyces.
Oh, streptomyces. Okay.
Yeah. Okay. Gene therapy is being used for tuberculosis, hematopoietic diseases, diabetes, and hepatitis.
That one is correct.
Isn't it getting used for diabetes because of insulin production? I don't know. I
Yeah,
don't
But
know.
I
gene
think
therapy means
metal,
you have to replace
Treating,
some.
yeah, like
You have to
using,
replace.
yeah.
yeah, you have to alter some genes.
Okay. All right.
Hiyo swali ya gene cloning vectors. Naona bacteria nini is also a gene cloning vector. A bacteria artificial chromosome is also a gene cloning vector. Well, according
What's
to
So
a
ni
the
bacteria
bacteria?
phage lambda.
no, even that is also
Actually,
a gene cloning vector. They are all gene cloning vectors. I don't know the answer.
the types of the types of vectors ni kuna bacterioplasmid vectors, kuna bacteriophage vectors, cosmid vectors, expression vectors, bacteria artificial chromosomes, na yeast artificial chromosomes. So I'm thinking all the answers that are there, or all of them are vectors.
I guess we can go with the
So
one that's
you go with
not
which one?
used
Yeah, I'd
most
go with C.
commonly.
Ningekufa na C.
I think I'd go with one that's not used commonly. Ama? I don't know.
Yeah, maybe, yeah.
Okay, so I guess we move.
Definitely.
All right, so the introduction of new DNA into germ cells by its injection into the, what the hell is this?
Nucleus.
Into the nucleus
Oh,
is termed as.
Is best
Oh, it's best.
it's termed as, yeah.
I think iko sawa trans.
Transgenic. All right. The following is true. Okay, we've done this.
Was it?
Okay,
Yeah.
but isn't this physio? Okay, ATP production takes place in the intracellular organelle. This guy knew his answers. So, which of the listed reagents may be used as a quantitative test for alpha amino acids? All I know is ninhydrin turns purple and is used for DNA. That's all I know. What do you guys think? Or number 13? Guys, am I here alone?
Sorry,
13. Which of the listed reagents may be used as a quantitative test for alpha amino acids?
Oh, I honestly don't know that one. I can't even remember those tests.
Quantitative test for,
It's ninhydrin.
okay. All right, so most protein
What are the
structures
other
that
tests used for?
question, yeah. We can always Google search it.
Yeah. Which one are you looking for? I look for another one.
Vanillin,
Vanillin. What was the
yeah.
other choice?
Dragendorf
That second one is for alkaloids.
alkaloids.
Oh, alkaloids.
Yeah.
Okay, so the other one is No? Failings.
Failings.
Failings. I remember our failings. Yeah. It's for, oh yeah, it's the one for reducing sugars and non-reducing sugars. The way they turn yellow, the one.
I think for vanillin, according to what I've seen on Google, is aldehyde test.
Oh, aldehyde test.
Yeah.
Okay.
Okay, so
So,
most protein structures are stabilized by all the following weak bonds except hydrophobic bond, disulfide bond, hydrogen, and electrostatic bond.
mm,
I know. So, all the following weak bonds.
but I don't think the sulfide bond is it a weak bond? Because it's a covalent bond.
Neither is hydrogen bond really a weak bond. I don't know.
Yeah, I think some hydrogen bonds are, but I
But it's asking for most protein structures as stabilized.
the following weak bonds except because okay, I'm seeing here like when you break the disulfide bond, it's an irreversible protein denaturation occurs. So I think it's a strong bond. No. Yeah, I'd
So
have gone with the sulfide also because I'm seeing it's a covalent bond, so it's not a weak bond. But I don't know, I'm not sure.
right. Okay, so we move.
Yeah, we can move on, we can start it. Yeah.
All right. Which of the following statements is false with reference to enzymes? So enzymes are protein catalysts for biochemical reactions. There is measurement of enzyme activity is used in disease diagnosis. Enzymes are reaction-specific catalysts, and each enzyme catalyst catalyzes several similar biochemical reactions. Catalyzes. Don't some also break, as in like lyse, like break it? I don't know.
Yeah, but I think like for a reaction, it can be maybe they're joining to form something or they're breaking down something. So it can work either way.
Either way, right?
Yeah. So each enzyme catalyzes several similar. So if they are reaction specific, then they can't catalyze several similar biochemical reactions. So it has to be one of the two.
Enzymes are protein.
But I think D is correct.
Okay, I guess D, and then we'll come and look at it. Is Gaius around? Or anyone else for that matter?
Hmm. Few
Okay.
enzymes exhibit absolute specificity. Other enzymes will be specific for a particular type of chemical bond or function. So yeah, D is the answer.
All right.
Mm.
So we move on. We have
Mhm.
15 more minutes. So, which of the following vitamins is not an essential constituent of coenzymes? Cyanocobalamin, I think that's thiamine, yeah.
Yes,
Niacin, yeah. Cobalamin. Okay, from a video I saw on YouTube, niacin and thiamine is already there. So, I don't know about cobalamin. Cobalamin is a B12, right? Vitamin
Mm.
B12. So I think even that one is there.
So
Coenzyme,
should be.
ascorbic.
Mhm.
Yeah. Okay.
Mm.
All right. Dash is not an end product of amino acid metabolism. Ketone bodies, carbon dioxide, urea, and cholesterol. I think cholesterol will be in lipid,
Yeah.
right? Okay.
Mm.
What the hell? The pancreatic I think islet. What the hell is this?
Can't even see the what?
Pancreatic. I think I
iLab. iLab.
islet.
Maybe need the islets.
I think
Islet hands, yeah, islet or something. I
islet
anyway.
secretes all the following hormones except there's insulin, secretin, glucagon, and I think this is somatostatin. What?
Somatostatin. So it's secreting.
Yeah, that one.
Yeah.
So the pancreatic produces the following except
Like it's secreting.
secretin is produced in the
Up here to duodenum.
Right. Okay, guys, since you're already here, we have highlighted 14 and 15. Thoughts on it? Care for your thoughts? Did he just do?
No.
He's reading the question.
Okay, I'd also go with disulfide. Cause I don't think disulfide is a weak bond.
Mm-hmm.
Because see, disulfide is even what holds insulin together. So I don't think it's a weak bond.
Okay, please number 15.
With elimination, I'd also go with C.
D.
Because C is
Right.
definitely correct. B is A is also, yeah, with elimination wise.
Okay, so we move. All the following are examples of bile pigments except stercobilirinogen, urobilinogen, and stercobilin. Mesobilinogen.
I don't think I've encountered mesobilirubin. Mesobilirubin.
Let me just see.
I'm not getting a lot of information on this. So
I think it's supposed to be C.
Meso?
Yeah.
right. But I have also gone with Meso. I've never heard of it.
Never heard of it, right?
Yeah.
Yeah, mess stuck because all these others they are formed. It's Billy. It's meso.
Mm.
Okay.
And then
So I guess we'll just start it. We see it when we read further. Okay, so dash is the end product of glycolysis under anaerobic conditions. So that's lactate. It's anaerobic.
Yeah.
And then all the following are processes of liposomal protein synthesis except
So see, all those three are involved in transcription.
yeah.
But sasa see we can say nini na A, B, and C are all connected. Initiation, elongation, termination. Them they are one thing. So with elimination, you can just choose transcription.
Transcription.
Yeah.
Okay.
Okay, so we move next.
Yeah, strepto. Anyway, microbiome. Maybe.
You want to?
No, I've just seen something with some bacteria there.
Yeah, that's microbe. Yeah. Okay, so
Maybe Patu can answer this for us because he did nuclear pharma.
Stage of which one?
no, the site of protein synthesis. Guys, what are you talking about?
Yeah. Patrick wanted to answer this one.
Anyway, let's continue.
Okay, so the site of protein synthesis is, Patrick, do you want to answer? I don't think he's in the meeting.
Yeah.
Okay, so?
That's it. We have ribosome.
Yeah, I'd also say ribosome.
Yeah, me too.
I think it would help if for these questions we analyze the other choices, because maybe this time they ask that, now next time they'll ask something different. I think it would even help us revise the concept we haven't revised. Just my opinion.
Yeah, we can.
Okay, so mitochondria. So like analyzing, in this case, like we say the functions or what happens.
Yeah, like in relation to the question. Like why not? Yeah, like something of the sort.
Okay. So
I feel like mitochondria is energy generation.
Yeah, mitochondria, we have ATP. the endoplasmic reticulum. Now there we can say it's more of packaging of the proteins.
So there's the smooth and the rough.
Yeah. But
So
the
the
rough one
smooth,
is also involved in protein synthesis.
yeah,
Yeah.
the smooth one is the one that's mostly involved with the storage.
Yeah, storage, yeah, and package, yeah.
Yeah, and
Into
packaging.
them too. We used to call them vacuoles, right?
Yeah, vacuoles.
Or
Vacuoles ama
vacuole.
vacuoles, yeah.
Usi unapumua hewa mingi ukisema hiyo word.
And the nucleus.
The
That's the
nucleus, we can say
nucleus.
huko ni DNA synthesis, yeah. Yeah.
Okay.
I
Alright.
think, Divya, you've written it the other way around for rough and smooth.
I feel like rough is right because it has those
The ribosomes, like
yeah, the ribosomes on it. So rough is used for protein synthesis.
Okay.
Yeah. So apoptosis is of course programmed cell death. RNA differs from guys, do you have something to say?
What happens in the cytosol?
cytosol.
I think what
Cytosol take on a function. Maybe glycolysis or
cytosol.
Hmm?
Yeah, glycolysis. Yeah.
So pia, pia unaweza yield ATP kwa cytosol.
Oh
through other
yeah.
pathways. Yeah.
It's just that maybe we can say ATP is the main mitochondria is the main site. But
cytosol sio different na cytoplasm, right? Mama? They're different.
So must study fulani yenye inasema different.
Inasema ziko different.
Yes, Chai alishare some study with me.
Oh.
Allah sana.
All right. RNA differs from DNA in that it's a single-stranded, has thymine instead of no. Yeah, I'd go with this. Out of elimination, this is DNA, this is DNA. 2-deoxyribose instead of ribose, yeah.
Yeah, all the others.
Single-stranded.
And then mitochondria, ribosomes, and lysosomes are considered to be organelles. Energy in a cell is produced in the mitochondria, majorly. And then all the following are antigen presenting cells for helper T lymphocytes. I think this is immuno.
Immunology.
Yeah. So I guess we can go because the rest are also asking for the same. What is this?
Okay.
Innate. Cytokines. Okay. Let me move if you guys are fine. Okay, here.
Glycogenolysis.
Diabetes insipidus is associated with, what is this?
Profuse.
Profuse sugarly urine occurs due to lack of vasopressin, can lead to hypoglycemia. Is a disease of children only.
So for insipidus is the ADH, lack of vasopressin.
Okay.
Yeah.
And then glycogenolysis occurs in the kidney. It's inhibited by insulin takes place only in the liver is stimulated by hyperglycemia?
Okay, using it's uh stimulated by hyperglycemia. That one is out because it's stimulated by hypo, right? That's why we
Hypo.
wanna use glycogen.
Mm-hmm.
Yeah. Then it's inhibited by insulin. Yeah, that's right. Because if insulin is high when there is hyperglycemia.
That's so good.
So
Mm.
yeah, it takes place only in the liver. Now for this one, we didn't get a chance to look at. I think is it the kidney chai that or the muscle? There's one other place that also glycogenolysis can take
See
place.
muscle? I don't know. Yeah, muscle. Because kangekuwa kidney pia basi A
Yeah,
ingekuwa correct.
muscle basi occurs in the kidney. Oh yeah, true. Yeah, the muscles.
Okay. All the following are functions of Sertoli cells except nourishing developing sperms, secreting protein hormones. I don't know what this is. Inhibin. Inhibin.
Inhibin.
Let me just. Sorry, guys, I'm making me suffer. Okay, so phagocytizing defective sperms. I don't know, production of sperms. Except secreting protein hormone.
What's the inhibin? Oh, which is that word?
This one? Nourishing.
No, that hormone. Yeah, it's inhibin hormone.
Okay.
So it's a protein secreted by Sertoli cells. So that one is correct. They nourish.
But this one will do it under physiology. So
Oh, so we can just move.
yeah, we haven't covered it yet.
What about this? Do you guys want to go through this?
That's production of cell in the stomach.
It's also physio though.
Yeah, it's also physio. Yeah. We can just go on. We focus with the bio.
Okay. If you guys see any, please let me know. Oops.
Biosynthesis of cholesterol.
It's that polythene, yeah.
Ornithine.
Yeah.
Oops. Okay. Non-absorbable switches. Nope. Human insulin is produced by recombinant DNA modification of
E. coli, yeah.
And then
Devious, you answer the ones for statistics for us. You know, you publish. You know these things.
I'll sit down one day and I'll answer them all for you. Don't worry. I'll send you the answers.
How sweet.
Insulin over dosage. Nope. I feel like this is.
I think we're done.
I guess we can stop here. It's already 10:05. Or what do you guys think? You guys
Yeah,
okay?
stop.
All right.
Thank you, guys.
Thank you too. Good night.
Good night.
Night.
yani tu kazi yake inakuwa tu kusema good night.
Smoky ni nani?
Or you can just what's that thing? You leave the app and come back on.
Yeah?
No, not like close it. Like just leave it and come back.
Good night.
Just do it.
Kazi yako ni kusema tu good night.
Of course.
Mazee.
Uko
Aliongea, but aliongea once. Janet aliongea once.
twice.
Ai, hakuna cha twice. Aliongea once.
Twice.
That's good. That's progress.
An achievement.
An achievement.
You know it, you know it.
Ati umesema nini?
Yeah?
Ni gaya tunaongea